Hi everyone, I'm Yuan Li, founder of BioPlus Partners. Over the last decade, China's biotech has grown from a newcomer to a global player. On this channel, we will share the stories behind that journey and what's coming next. Stay with us and enjoy.
Hello everyone. Today we are in the center of Zhangjiang, Shanghai, and then we are with the CEO of EpimAb, Dr. Chengbin Wu. And then EpimAb is an established clinical stage company in China developing antibody therapies for multiple disease indications. So we are very happy here today to have Chengbin joining this talk. So Chengbin, could you please first introduce the founding story of EpimAb?
Sure. So hi, my name is Chengbin Wu. I'm the founder and CEO of EpimAb Biotherapeutics. So I think, I was the CSO of 3S Bio before I founded EpimAb. Mm-hmm. So around 2015, 2016 timeframe. So at that time, the Chinese biopharmaceutical industry just started to take off, and there's a lot of biosimilars, but nobody really understands bispecifics. So I thought that was a big unmet medical need for the biotech industry. Actually, you have to have a good technology to develop and generate bispecific antibodies. And this is also based on my personal experience. I actually worked with AbbVie for over 10 years in Worcester, Massachusetts, and I started work on bispecifics starting 2003. So it was by the time I started EpimAb, I have about 15 years of experience in this field. So I thought that was also my expertise that I could contribute to building and funding this new company and really provide contributions to the Chinese biotech industry.
So at that time, what are people's planning for the indication or application of this platform?
Yeah, I think initially there was three different major areas for the application of bispecifics. One is the tumor targets, dual tumor targets like c-Met EGFR that are both expressed on tumor cells, and this has been a lot of discoveries in that area, including like HER2, HER3, EGFR, all those kind of conventional tumor-specific antigens. The second area was at that time was just starting, which is immuno-oncology, right? PD-1, PD-L1 story. And people have seen that in the first generation of immuno-oncology drugs, there's acquired resistance in patients in the clinical setting. So people were thinking if we have a different checkpoint inhibitor in combination with PD-1, which could provide additional benefit or deeper response to patients in this area. So that was the second concept, which is the PD-1 or PD-L1 plus bispecifics. One of the successful stories is obviously PD-1 VEGF, right?
Yeah.
So the third and most conventional concept or mechanism for bispecifics is the T-cell engager, right? That's the third category, which is also the major category of bispecifics in clinical development these days. About 50% of the bispecifics actually in the TCE category.
Right now or back then?
Back then was less. Right now it's actually increasing over time. And this is also most proven mechanism for T-cell engagers. So I think in this particular aspect, the T-cell engager mechanism actually combine the targeted therapy mechanism with the immuno-oncology mechanism. So in this particular modality, you have a very clear target tumor cell type, right? And then you also engage the endogenous immune system, which is engaging the T cells through the CD3 binding to really activate the immune system in this very specific, target-specific way. So I thought that was a very interesting and also an important application for bispecific modality in general. And we actually have focused a lot in this area in the recent years.
Yeah, and I saw that those focuses already lead to two, very significant and successful deals with the other biotechs in the U.S., right? So there must be a fantastic story behind it. So can you talk us about this deal?
Yes. So that was also the first TCE project we actually started in-house, which is the BCMA CD3. So along the process of developing this program, we realized that the technology platform that we have developed in-house, which is called FeedIG technology, is very suitable for developing T-cell engagers because of the many of the unique properties of the FeedIG architecture, meaning it's a bivalent, tetravalent bispecific formats, and the distance between the two binding sites are very close, so that you don't need to use very high affinity CD3 in order to engage effectively and provide a very potent tumor killing activity. So this is actually fully realized in the clinic with this first program, where we saw over 90% ORR in multiple myeloma, but very limited CRS observations. So this is actually a very interesting story of how to predict your technology advantage, going through all this translational process and really finally realize all of your predictions in the clinical setting. So I think when we started BCMA CD3 program, we were not the first one. But when we look at all the data, preclinical data, including monkey data, we were convinced that we have a best-in-class molecule, right? And which has a very differentiated safety profile, particularly in terms of low CRS in the monkeys. Okay, so and then that's exactly what we saw in a clinical setting.
When is that?
That was about 5 years ago.
5 years ago.
Yeah.
Okay.
Yeah. So this program, we complete the full phase 1 program with the full range of dose escalation from very low 2 mg all the way to 300 milligram, to demonstrate this molecule is both efficacious and also very safe in cancer patients. And we've done a lot of biomarker studies in the clinical setting, and we've looked at the disease parameters in the bone marrow, and also we look at B cell depletion and IgG reduction in peripheral, and we realized that this BCMA CD3 can effectively delete or deplete multiple myeloma cells, plasma cells, plasmablasts, and also peripheral B cells. So that is very interesting observation, and this is actually a very important basis to move this program into autoimmune indications, where the B cell depletion is a major mechanism for many of the autoantibody diseases in the autoimmune space. So this is the first program we completed in the TCE project, in the TCE space. And conversely, this project also validated our technology platform to further enhance or further advance other TCE programs into autoimmune and tumor programs.
Yeah, so then we all know that deal quickly evolved into a merger and acquisition by, Foresight. Foresight
Yes.
are behind those deals. And everyone knows, TCE asset from China. I mean, that's like opens the door for a lot of, projects after that. And instead of, there are all different kinds of ways, when you have a validated platform in your hands, right? There's all different kinds of ways of strategies that you can develop it.
Yes.
And then we see that very soon in this year, in May, there's another deal, right? On another target, KLK2. I guess that's not a target everyone talks about, every day, right?
Yeah.
So instead of, so how do you think of the strategy of the company? How do you make the choice to kind of out-license the second asset or to develop by yourself? What's your strategic thinking out in this?
Yes. I think the KLK2 project very uniquely demonstrate our discovery strategy, which is more into the first-in-class, or one of the first-in-class discoveries and developments in the TCE space. So we actually look at all the targets for solid tumor initially, and identify those that are very unique and appropriate for TCE development. And some of the targets are better for ADCs, but we have identified targets that are better targeted by a TCE modality. And also there's a large unmet medical needs, and obviously potential interest for deal makings with big pharma. So KLK2 emerged as one of those targets. And this is actually a very tough target to work with. It's not a conventional receptor. It's actually a secreted protein, but somehow reattached to tumor cells in prostate cancer. So early discovery was very challenging. We actually worked on this for over 2 years before we reached the PCC stage. So this is a very good example to demonstrate how we look at discovery, not really to get into a very crowded area to follow everybody else, but also to instead to really identify an area of interest that can take advantage of our own technology platforms, our know-hows and capabilities. We actually published, we had a poster at AACR meeting. We sent out the abstract. So the AACR first published abstract. As soon as the abstract came out, TCG Labs came to us.
Okay.
So they didn't look at the data. There was no data, right? So we signed the CDA, then look at the data. We actually signed the deal before ASCO when J&J published their data.
Well, that's quick.
Yeah, very quick. Yeah. They wanted to quickly close this and move on.
Wow, that's very impressive.
Yeah, we also think they are a very good partner. They have very good expertise in prostate cancer clinical development, and they have a very strong support financially and also technically to move this program forward.
This out-license deal is like they are in charge of the development of the product globally?
Yes,
or
it's a global partnership. Yes.
Okay. So looking forward for EpimAb as a company, and then I feel like now it's pretty established. It has its own technology platform. It has its capability to successfully, develop products not only by itself, but also recognized by the field, which is very important, right? And then for EpimAb, looking forward, how would you say the strategy of the company? Will we see more immunology pipeline, or how would you, allocate the resources in different disease areas?
Yes, I think the easier way to think about this is to repeat what's been working well for you. Right. So from the BD perspective, the 2 TCEs is one for autoimmune, which is BCMA CD3, which is going very well in the clinic right now. The other one is KLK2, which is solid tumor TCE. So this is actually the two important directions we're taking in our discovery to move several innovative programs forward. In the solid tumor, we have another innovative program coming up for IND filing toward the end of the year, which is the LPP CD3. So I think we will be the first in clinic for this type of asset. Along the way of discovery and technology development, we're also developing the Pro-TCE programs, the TCEs with masking technology, right? Either a single masking of CD3 or dual masking for both TIA and CD3. So we have two programs in that area that also move along very quickly, and they're all very important targets that express on multiple tumor types. Okay, so those will be following very quickly after LPP. On the immunology side, it's the same thing. We have a tri-specific B-cell depletion program, which is moving very quickly into preclinical development, which is a CD3 plus a plasma cell target we have not disclosed, but it's a very unique targets that are specifically highly expressed in SLE patients. more than BCMA, so it's not BCMA. Yeah. And we also venture into areas outside of the B cell depletion arena. So we're looking at other immune cell types that are involved in important immune responses, some of which are actually type 2 immune response, like asthma, allergy, those type of responses. And some of the immune cells are importantly involved in those processes. So we're targeting to deplete those type of immune cells with the TCE technology. So those are the major directions we're working on right now. And we're hoping to bring at least 1 or 2 new IND filings in both areas next year.
Yeah, I already heard a lot of, keywords that will make the, the field pretty exciting with.
Yes.
Besides what you just mentioned on the exciting, new discovery pipelines, are there any specific milestones you like our audience, like the investors or any partners, to keep closely watching?
Yes, I think definitely in a clinical setting, we're moving 2 clinical programs forward. Our Ep.1, c-Met EGFR, we're developing that for metastatic CRC. And we're also developing our 1 CD3, which is still in phase 1 dose escalation. So those are the important programs for us to move things forward. Our partner is Candid, is developing BCMA CD3. They are also moving very quickly into the multiple indications in the autoimmune space. They have like 3 INDs approved already for 3 different indications. For us, I think the next few years definitely would be important to further demonstrate our TCE molecules in solid tumors, as well as for autoimmune indications, both specific as well as mechanistic specific. So we wanted to actually further validate the TCE technologies, the Pro-TCE new technologies in the solid tumor space, because I think solid tumor is much larger market than the hematology. And also, as you mentioned, autoimmune is also a huge market. This is actually we're just starting, right? Last year was the first year we have a clinical demonstration of TCE, B-cell depletion TCEs in autoimmune indications. A lot of those studies are still very early studies with limited patient numbers. So we're looking forward to more validations of these type of mechanisms in autoimmune indications in the next couple of years, and hopefully with much larger patient population, also with a lot of clinical data demonstrating efficacy as well as safety and durability. So those are coming up in the field that are important for the next stage of TCE development.
I see. I think we have put a lot of emphasis on the technology and on the great pipeline that is like emerging, right, about developing inside. So I'm wondering, commercialization is actually another important piece of this. How do you fit the commercialization into the current map you're having in your hand?
Yes, I think this is a very important question for any biotech companies to consider. So for us, I think the strategy is really to partner. That's why we stress a lot on early partnership, as early as preclinical, but also in the clinical stage. We also very motivated to partner with multinational companies to really bring the product to the market together. With our focus is obviously in China, but I think EpimAb as early as the beginning of the company establishment, we always have a global mindset and product development as well as for intellectual property. So this is the area we continue to seek partnership.
Okay, thank you very much for joining us today, and then we'll be looking forward to the all the exciting deals and partnership in the future for EpimAb.
Thank you very much.